So welcome back for topic 6.6. We're going to talk about gene expression and cell specialization. So let's go ahead and I found this image online. I think it's a pretty good image because it shows me the basic things I want you to, sorry about that. I want you to understand about how eukaryotic cells in particular kind of regulate a lot of their expression of genes. And there's many different factors to this. It's much more complex than this, but this is kind of a simplistic image right here. So some of the things I do want you to know is that genes, right? If I have a gene here, let's put my gene in red, right? This is when I say gene, what I'm talking about typically is the specific region that will be transcribed, right? This is what's going to be generally transcribed into pre-mRNA, right? You're typically going to have at the beginning of a gene, right? You're going to have something called a promoter sequence. Promoter sequence up at the beginning of the gene that is going to act to be kind of the area where RNA polymerase can attach and begin transcribing, right? Now, as you're going to see here, and this is, let me use a different color here. Let me use yellow. Sometimes many thousands of hard to see the yellow cursor. Sometimes many ways away from the gene, very far away, you're going to find something called an enhancer region, and I'll put that region in even a different color. Let's put it in green. Green for go. An enhancer region on a gene. And what's going to happen is you're going to have a couple of different types of proteins that are going to bind to these, either the promoter and the gene. To the promoter, oftentimes what you'll find bind is these things called, let me change my color back to blue over here. Maybe I'll make it light blue for protein. You're going to have these transcription factors, right? Transcription factors. I'll put TF. This is something you do need to understand is the basic function of a transcription factor is to affect gene expression, right? Transcription factors will bind to the promoter. Oftentimes you'll have something also called an enhancer. This is not always the case, but in many cases you'll have some sort of enhancer protein here, which will have something called an activator protein, which can bind to the enhancer. Activator protein, right? And what this is going to cause is a bending of this DNA and an activation of this gene at a high rate. So enhancers, as you might guess, enhance mRNA production. Enhancer region is to enhance mRNA production. Sorry. The promoter kind of allows this RNA polymerase, which I will put in, let's see, purple over here. Right? This is our RNA polymerase. To attach and go ahead and transcribe this gene, right? Now, this enhancer region oftentimes will say, hey, I need this, I need a lot of it, I need it quick. It's going to kind of come loop around here and kind of act to enhance the production of this gene, right? As you'll notice, you have your RNA polymerase down here, which is getting ready to transcribe in this leftward direction right here, or as they call it, downstream, right? This direction is downstream. Let me write that a little bit better here. Downstream. And of course, this direction in relation to a gene is upstream. And this is just general terminology to discuss which direction you're looking at when it comes to transcription of a gene. So transcription factors bind to your promoter to enable this RNA polymerase to start transcribing. Oftentimes, these enhancers, which can be located thousands of base pairs away, right? These enhancer regions on the DNA right here, which have activators that can bind to them, will cause the DNA to loop around and cause increased transcription, right? Increased transcription of this gene. Now, there's many other proteins involved, but this is a high school biology course, but there's probably even proteins involved in the bending of this DNA, right? Causing this DNA to bend around and things like that. So there's many more proteins involved. It's typically not this very simplistic process you might look at here, but I do want you to understand the basics of how transcription works in a eukaryotic cell. You'll also notice out here in this image, they have these methyl groups attaching, and it talks about insulators. Don't worry too much about insulators, but do know that sometimes these methyl groups, as we talked about when we talked about epigenetics, can silence a gene and cause it to not be transcribed, right? So first of all, let's take a look. These are, I'll let you take a guess. What do you think these are over here? These are actually E. coli, Escherichia coli bacteria, right? And these are things that exist in your stomach. You have a ton of them, and they break down many different types of food. So if you eat some protein, these E. coli have to break down protein. They help you break down lipids. They'll help you break down sugars, right? One of the particular things we're going to look at is something called the lac operon, right? Lac is short for, lactose. Lactose in this case, or it's talking about the gene relates to lactose, which, as you know, is a disaccharide. And the word operon, this is a group of genes near each other which help to regulate the synthesis of proteins. So E. coli, in order to save from making stuff to break down stuff when they don't need it, right? If you have an E. coli bacteria and you don't have an influx of protein, you don't need to be making enzymes that break down protein. So how do they kind of make sure that they're making the correct enzymes at the correct time? And instead of, kind of distributing, I'll have 20% of my enzymes this one, 20% this one, 20% this enzyme, 20% this enzyme, and 20% this enzyme, in which you get kind of mediocre activity no matter what happens. So it has kind of a genius way to say, hey, I need this right now. I'm going to make a ton of this protein if this is to break down something. Or I'm going to make a ton of this protein to break down a protein. Or I'm going to make a ton of this protein to break down a lipid. In this case, we're going to talk about lactose and the presence of lactose. It's a well-studied operon or group of genes. And instead of showing you that, I'm going to kind of do a little simulation here that you guys are going to do at home anyways. So let's go ahead and take a look at a little simulation that I have lined up for you guys to do on your own. Okay. So let's go ahead and take a look at what we've got going on here. This is, we're going to talk about a little bit about lactose regulation and the lac operon. So first of all, you're going to look down here and you're going to see this lac-I promoter, lac-I gene, lac-Z gene, and lac operator. Don't worry too much about those specifically right now. Let's see what they do. I'm going to go ahead and add this. I'm going to do this one first, the lac-Z gene. I'm going to put the lac promoter over here, and I'm going to put the operator. So as you'll notice, can you guys guess, first of all, what this enzyme is based on what it's doing? This is an RNA polymerase. It is transcribing some mRNA, and that mRNA, of course, is going to become a protein. Now, we don't know what that protein does yet. Just know that it's making a certain protein. This lactose injector popped up over here. Let's go ahead and inject some lactose. This is lactose, a disaccharide. Keep in mind, this has to be broken down into glucose before it can kind of enter our system. So have you figured out what this protein does that we're making here? You should have guessed that it breaks down lactose. Now, this, of course, this is some DNA inside of an E. coli somewhere, right? It's constantly making this protein to break down lactose. But as you'll notice, right now I'm not making any lactose. So is it very useful to be making this long, large, complex protein that breaks down lactose? Probably not. So you have another gene over here with another promoter, right? This one over here is going to transcribe something else. Now let's see what happens here when we see this polymerase kind of move that direction. I'm going to speed it up just a little bit to save you guys some time here. It's going to make a different protein. This protein is going to bind on the operator, and let's see what it does. You might be able to guess from here. Take a guess if you want to pause and take a guess right now. It's now blocking transcription of this LAC-Z gene. By blocking transcription, and you're making this small, simple protein which binds to DNA, you're keeping this E. coli from making this kind of useless protein, right? This protein's useless while we don't have lactose. Well, again, let's see how this works all together. I'm going to inject a bunch of lactose here. Let's see what happens. Take a guess as what you think is going to happen. Notice that this lactose will bind, right? This lactose is binding with this protein, this repressor protein, right? Which is the repressor protein is something that binds to that operator. Once the lactose binds, it changes shape. It cannot bind to the operator, and you're going to see transcription again of this gene so that it can break down all this lactose. So let's see what happens here. We made our first one. Let's go ahead and make it go a little bit faster and see what happens. Now we're making even more. But again, I have a finite supply. I don't have it on auto up here. It's manual. And all this lactose is now broken down. So what do you think is going to happen? Well, these proteins here are going to get broken down to their component parts eventually. And there's not going to be many more of them made because you don't need them. Cells can, an E. coli cell can go from having none of this protein to having a lot of its biomass consisting of that protein. Almost half of it can be this protein, this protein which breaks down lactose in the presence of lactose. So when you take a gulp of milk, your E. coli are going through this very complex process and making certain genes very quickly, or making certain proteins very quickly so that they can break down that lactose that you've fed them. There's also a lactose transport module over here that I'm going to let you do on your own. It is very similar. The only thing it's added is a protein which will embed itself in the cell membrane in order to allow lactose in. So that's the basics of the LAC operon. I'd like you to play with this. I put a link up to this in Schoology, so you should be able to play with it. And let me get back into our slides here. Should be able to play with it and see kind of how the basics of how an operon works, and how it kind of operates to either repress itself or generate a protein to break down a specific molecule. And I think I'm going to leave it at that for this video. So I will talk to you next time.